Assalamu alaikum. Hello guys, welcome back to my channel. Today, uh, after finishing epithelial tissue, we will go to connective tissue. That's one of the most important tissue types in our body. That's more complicated actually than epithelial cell. There are a lot of types of cells. For example, we have two types of cell: fixed and permanent cell. I will explain those in other videos. Okay, the types of macrophages and other cells, but now we will just see just like a general feature of connective tissue, general information. They are derived from the mesoderm, however the ectoderm of the head region may also contribute their formation. So as I explained, the upper part of any, as I gave you some slides in my past videos. In every slide, for example, you have a skin slide. Okay, a keratinized skin slide. We see the keratin at the first. After that, we see the epithelial cell. How to identify a connective tissue? Under the basement membrane, which stains blue, the basement membrane I showed you in other videos and slides. If you go to epithelial tissue slides part 2, you can see in the epithelial tissue slides video, you can see what's the basement membrane and learn about it. So under the basement membrane is the connective tissue, and the connective tissue is more loose than the epithelial tissue. The epithelial tissue is attached together by junctions, okay? So second point, they are well vascularized. So there are a lot of vessels in the connective tissue. But we may have different information, such as when we reach cartilage, in the outermost layer of the cartilage perichondrium, we have vessels, but in the cartilage cells we don't. I will explain about them in other videos. So they are well vascularized. They are made of three components, okay? Fibers, cells, amorphous ground substance. I explained about fibers in my past videos: collagen fibers, elastic fibers, and the types of fibers, and reticular fiber. So how to know them and how to identify them, I did explain about it. We will come to function. We have some main types of function, depending on the way that the connective tissue has a role of in our body. So first we have mechanical function. What's the mechanical function? A, it supports. B, it causes connection between for example, muscle and bone. Protection. Okay, we have body defense. Number two, body defense. How they defend body? By the presence of first A, macrophages, phagocytosis. Okay, so what are the phagocytes? They engulf uh, pathogens. So what are the pathogens? Bacteria, viruses, and fungi. So they have sometimes yeasts. It depends on the type of infection. The phagocytes, they engulf the pathogen. So presence of immunocompetent cells. Okay, and physical properties of ground substance acts against spread of pathogens. Number three, we have storage. So connective tissue stores waters and electrolytes, such as NaCl, sodium chloride, okay? B, fat and adipose tissue. So fat is stored in adipose tissue. And for example, the main place of fat storage is our liver, okay? If we stain a liver section or a liver slide by osmium stain, we will see a lot of adipocytes, okay? Temperature regulation, such as adipose tissue. We know that simply how to recognize that. People that are obese, that have obesity, their body is much hotter or much warmer than people that are thin or skinny, okay? So adipose tissue regulates our body temperature. So tissue repair by formation of scar tissue. We have phagocytosis simply. So what's phagocytosis? Okay, if we come to this figure, we see a cell. So we have, in every cell we should have nucleus, or in some cells we have more than one. We have a cytoplasm, a cell membrane, and some organelles. It depends on the type of the cells. Okay, so what we have here at our first stage, we see, for example, a food particle or something pathogenic in our body. So here, the plasma membrane forms like a cave, okay, to engulf the food particle, or if it's a leukocytes engulf pathogens, or white blood cells, as I say, phagocytes. They engulf pathogens. So here you see the membrane just forms just like a cave, and lysosomes we can say it gets active. So why lysosomes are, we know that they are, I explained in cytology videos, lysosomes are digestive organelles, we can say. They have digestive enzymes that digest food and dead materials in the cell, okay? So the lysosomes, move and get ready, we can say. So in the second stage, as you see, the cell membrane is much tighter, okay? It's near to engulf the food particle, and we come to this stage. We see that the particle is engulfed. So the fusion of lysosome with food vacuole. The food vacuole will be formed in this stage, okay? Food vacuole will be formed. The particle will enter the food vacuole, so the lysosomes will fuse to the food vacuole. What means fuse? Fuse means they attach to the food vacuole. So if we just, I will try to draw a line on the food vacuole. So here we have the food vacuole, as I mentioned. Those small granules are the lysosomes. The lysosomes attach to the food vacuole to start, and after that the end stage is digestion. So the digestion will occur by the lysosomal enzymes, okay? And the cell will use the food particle as a source of energy, okay? Here, let's take a simple case, a very simple case that's important to know. For example, you have a dog, a canine patient, with a lump, okay, with a cutaneous lump, okay, that you don't know if it's, what type of lump is it, what is it? Is it a tumor, neoplasia, or neoplasia is cancer, that I will explain about it when we in my other playlist, reaching animal diseases. Neoplasia, is it an cyst, an infection cyst, or is it, what type of tumors? Is it round cell tumor? And the round cell tumors are divided into many types. So simply, for example, as a vet, you had a case of a skin lump, unknown skin lesion lump, or something like that, okay? You can't diagnose it. Never, never, ever. You can't say this is for example, this is cancer by just only palpating the lump, or by just seeing the lump. No, some other tests should be done. First thing, of course, doing some CBC, okay, hematology tests, CBC, and aspiration is taken, FNA, fine needle aspiration. Fine needle aspiration is a technique which a vet, or sometimes a physician, or it depends on the nurse, or it depends on the place and who will take the sample. Mostly vet doctors and physicians will do it, okay? A needle is inserted to the lump, as you see, and you know the air is pushed back to create a pressure, okay? And you will move back and forth quickly, okay? Just like something like that. You will move, I will explain, about 15 times back and forth, okay? So how is it done? Of course, when you enter the needle after disinfecting the area, you will move just like that, about 15 times quickly, and aspirating with it, okay? You aspirate and move just quickly to the muscle like that. It means you create just like injecting several times. So you will get a drop of fluid and you will put it on a slide. After putting it on a slide, you will spread it. So I will learn you how to spread, how is cytology smear done using some other things, not animal tissues, just for training. Okay, after staining it under the microscope, you found those cells, okay? Those types of cells. I wrote 2 images here. As you see, those are the red blood cells that I will explain about in other videos. Those ones are the red blood cells. So yeah, they stain eosinophilic. I explained what's eosinophilic and basophilic in my other videos. Here we have a rupture. That's actually not something abnormal because the smear is done, maybe something, maybe a rupture is done because of the smear. Rupture is occurs because of the smear. This one is a rupture here, okay? Here, the strange thing, we know that here we can see phagocytosis. So here in this image, you can see a bacteria here, as it's mentioned. This is a bacteria. It's engulfed by a neutrophil. Neutrophil is a type of white blood cell. Okay, it reacts when inflammation is there or any infection types, mostly bacteria, fungi. Okay, this type of white blood cell will be activated and engulf the pathogens. So here we can see a bacteria near a neutrophil, and here we can see a phagocytosized bacteria. Sorry. Here we can see a phagocytosized bacteria in the cell. So that means the cell has been, that means the presence of infection, bacterial infection. Here we can see other bacteria in the cell. This is a neutrophil cell, okay? This one is neutrophil. When you see this case in a cytology exam, that's actually abnormal. That indicates inflammation. So, as you see, a bacteria here. This is phagocytosis under light microscopy of cytological examination. So here is the bacteria, this one that I mentioned. It's phagocytosized by the neutrophil. So this one is the nucleus, that basophilic bilobed nucleus. This is the neutrophil has a bilobed nucleus. It consists mostly about 3 lobes. Okay, so hope you get benefit and understand me. Here, before I end the video, we have another example. This is a phagocytosized bacteria. You know, the video is so a little bit blue, I don't know. So this is the simple explanation about phagocytosis. As I said, the neutrophil or any type of white blood cell engulfs the pathogen. So the pathogen is here. This white blood cell, as you see, the membrane will will make it just like a cave and attach to the bacteria and engulf it in the bacteria, and the bacteria will be killed by the neutrophil. If you see a microscopic image like that, again, that's actually abnormal. Be careful. That indicates inflammation. That's not normal. If you say, okay, that's normal, just a phagocytosis. No. Phagocytosis of bacteria indicates inflammation. So, thank you.